Hello. So today we're going to be continuing our conversation of the cell and cellular biology, the study of the cellular structure and function. And we've been talking about our plasma membrane, and we concluded that conversation. The next aspect of our cell that we'll be discussing is the cytoplasm and the organelles. So our cytoplasm. The cytoplasm is roughly everything inside of our plasma membrane. So our cytoplasm includes these little structures called organelles and the fluid that surrounds the organelles. Now, some resources are going to differ on whether the nucleus itself is an organelle, or is it its own special organelle, or is it something else entirely. We're not going to worry about that part. The main differentiation here that I would like us to capture and really integrate is that the cytoplasm is everything within the cell, including the organelles, including the fluid that surrounds the organelles, whereas the cytosol is just the fluid within the cell. It excludes the organelles. So we can think of this as those really weird desserts that tend to show up at summertime picnics, right? You know them, these little Jell-O desserts that have fruit within it. So we could consider everything within the boundary of this dessert. We could consider all of it the cytoplasm. Now, that cytoplasm is made up of the fruit itself, the solid components, or the organelles, and the Jell-O, the fluid that surrounds the fruit, the cytosol. Now, so that's the cytoplasm. Let's look closer at the organelles. So we've been talking about this idea that the entire cell is a kingdom, and that the plasma membrane is the boundary around the kingdom wall, right? Well, within any kingdom needs to be a number of facilities that help that kingdom to function. So there's transportation systems, there's postal systems, more transportation, factories where goods are made. We're going to see that within our cell, there's those same functions that need to be achieved in order for any particular cell to thrive. So let's look at those organelles. The organelles that we'll be talking about today include our cytoskeleton, our lysosomes, peroxisomes, mitochondria, ribosomes, endoplasmic reticulum, the Golgi apparatus, and then finally the nucleus. So our cytoskeleton is a lattice work of microfilaments and microtubules. So very small filaments or threads, and microtubules, very small tubes. And these help to maintain cell structure, just like a lattice in a garden gives some structure to the vines that are growing up it. And this lattice work provides for cell movements, so that proteins and organelles and chemicals can move within the cell. So if we look at this lattice work, we see that it's like a whole highway, right? Transportation routes, right within the cell. Different motor proteins are going to transport organelles, the nucleus and vesicles, and large molecules such as proteins and messenger RNA within the cell. So again, we can think of our cytoskeleton as little food substances, right? So it would be like the coconut in another version of that semi-disgusting Jell-O fruit. This one has whipped cream dessert, right? So the cytoskeleton here would be the coconut, that grainy, filamental structure. Now we can get a little bit more specific with our cytoskeleton, the skeleton of our cell. Anytime we see cyto, it refers to our cell. So here, the skeleton of the cell. And we'll look at the different types of cytoskeleton. So the first that we come to are our microfilaments. Now, our microfilaments tend to be made up of a subunit known as actin. So each of these little blueberry-like circles is an actin subunit, and the actin gets strung together to form these microfilaments. And the microfilaments perform a function of giving the cell structure and allowing for contraction. Now, on average, the diameter of our actin microfilaments are about 7 nanometers. And again, the protein subunit is the actin. And we can see that within our cell, here's the nucleus of our cell, which we'll spend more time talking about, but all of these blue fibers, highways, are our actin microfilaments. Next, we have our intermediate filaments, and our intermediate filaments tend to be very fibrous in nature. They're on average about 10 nanometers, and they form these large, sort of web-like branches, like a bat-like network within the cell. We also have microtubules, and these microtubules are very small tubes. So we can see and sort of appreciate the tube-like structure. You can imagine that something could pass through here or along here within these microtubules as they're oriented in a semi-helical fashion. And these microtubules, their function is to provide structural support, and they make up our mitosis spindles. They tend to be much larger in diameter, about 25 nanometers, and their main protein building unit is something called tubulin. So in this image, here we have our nucleus, and here we have our tubular structures extending out in these gold networks around the nuclei. So that's our cytoskeleton. Next we come to our lysosomes. Our lysosomes are like our trash receptacles, so a nice tribute to the classic Star Wars movie where they're trapped in the trash receptacles. And our lysosomes are analogous to that. So lysosomes are membrane-bound organelles that contain digestive enzymes, so enzymes that can break things down and destroy them. What we see is within a cell, there'll be primary lysosomes that fuse with vacuoles or other organelles, forming secondary lysosomes that degrade foreign material and cellular debris. So let's look at this. So here we have some sort of substance that is brought into the cell, and we form a vesicle. So here's our vesicle of something that was once outside the cell and is now inside the cell, right? So that vesicle is then going to fuse with our primary lysosome, forming a secondary lysosome. So our vesicle plus the lysosome again has all sorts of digestive enzymes in it. They'll fuse together, and those enzymes begin to degrade the material that was brought into the cell, into those vesicles. So this is the cell's garbage disposal system. It's a way of cleansing, of clearing out, of getting rid of anything that's harmful for the body. Now, these lysosomes also play a role in apoptosis, or programmed cell death. So sometimes cells need to destroy themselves, right? They have a little suicide clock within, and this comes into play usually whenever a cell is damaged. We don't want damaged cells proliferating and prolonging their life, and so cells will self-destruct. Lysosomes play a key role in that self-destruction. Next, we have our peroxisomes. Peroxisomes are fairly similar to our lysosomes, except that the enzymes bound within this particular organelle are going to be oxidative in nature. So we say that they're oxidative enzymes. So these peroxisomes form hydrogen peroxide, H2O2, by removing hydrogen from various organic molecules and transferring it to molecular oxygen. Peroxisomes are used for detoxification. A good example is in our liver, where peroxisomes convert ethanol to acetaldehyde. And H2O2, it's important to note, is actually toxic to the cell. So once H2O2 is formed, it doesn't stay around as long as everything is functioning normally within the body. And that hydrogen peroxide is broken down, ideally, by a peroxisome enzyme catalase. And this then neutralizes the system. So it destroys whatever was originally coming into contact with the peroxisome, and then there's a neutralization process that ideally happens. And next, we'll look at our mitochondria. So mitochondria are the sites of oxidative metabolism. So within the cell, there's these storehouses where fuel is made, where energy is made. So oxidative metabolism is a series of reactions that require oxygen to produce ATP. All right, so mitochondria make ATP. What's ATP? Well, ATP is the primary energy source for most cellular reactions in our cells, in our body. So it's a huge energy source. And ATP stands for adenosine triphosphate. So it's adenosine plus 1, 2, 3 phosphates. Adenosine, adenine plus a ribose group, adenosine, 1, 2, 3 phosphate. Now, ATP, again, it's our body's currency. It's our body's energy. So we might take into our body lipids or proteins or sugars, and those are energy, it's true, but our body can't, for the most part, can't utilize that energy until it converts it into a currency that it understands, into ATP. So we can think about this as doing laundry at the laundromat. We might go to the laundromat and it costs, $1.25, $1.75 to do laundry, and the machines only take quarters. That's the machine's currency. But we walk in and we only have a $5 bill. Well, that $5 bill is not going to get us any clean laundry, right? We need to use a little money machine to convert our $5 bill into quarters. The same thing is happening within our mitochondria. Certain energy, glucose, fats, protein, etc., are going to come into our mitochondria and get turned into ATP that our body can use for energy. Very important, these mitochondria. So now let's look at ribosomes. Ribosomes are the protein factories wherein the cell's proteins are synthesized. And these are composed of 2 ribosomal RNA or rRNA subunits and proteins, a large subunit and a small subunit with a little space in between, and that will become important in physiology whenever we talk about translation. So our ribosomes we can think of as these protein factories. They're making proteins building blocks of our body, right? We need proteins. These proteins are incorporated into our cells, into our organelles, into our tissues, all over. And it's our ribosomes that make them. Now, ribosomes can be either free in our cytoplasm, so floating around, and these generally are sites of protein production for use by the cell. So if the cell is going to use the protein, then generally it's made on ribosomes that are floating around in the cytoplasm. Or ribosomes can be bound to our endoplasmic reticulum, and these tend to be protein or for protein production for either export or for a specific organelle within the cell. So let's look at our endoplasmic reticulum. Our endoplasmic reticulum is a system of membranes specialized for synthesis or degradation of molecules. Now we have two types of endoplasmic reticulum. We have something called rough endoplasmic reticulum, or RER, rough endoplasmic